Hello, I'm Dr. Dabe, and I'm a bioinformatics consultant at BISC Global. We are, as our name implies, a global bioinformatics, statistics, and machine learning consultancy firm with offices in Europe and North America. And today I'm going to talk to you about single-cell RNA-seq. So whether you're just getting started with single-cell RNA-seq, or you've already processed some data sets of your own and are looking to optimize, automate, or otherwise expand upon your existing analyses, this is the video for you. In short, our goal is to help you make the most out of your single-cell RNA-seq datasets. So we'll kick it off with a high-level overview of the workflow, and then move into some examples of the quote-unquote standard single-cell analyses that you will commonly see in the literature. And then in the second half, move on to examples that we at BISC think should be part of standard analyses, including additional in-depth QC and cluster optimization steps, and expanded analyses including pseudotime analysis and gene regulatory network analysis. So this is our general overview of downstream single-cell workflow, whether you're looking at public data or in-house proprietary data. So regardless of what platform you decide to use, after you've sequenced your data, you're going to do some sort of read mapping against a reference genome so that you acquire some data tables that have your counts of gene expression per cell. Once you have these data tables, you're ready to start. So we first want to homogenize our data. So we're going to perform some quality control, including removing any dead cells or any duplicated cells. We can perform normalization on the counts data for each sample, and then we can merge multiple samples together into one single cell cluster diagram. So this is going to require some degree of batch correction and cluster optimization. Finally, we get into our differential expression and differential comparisons. So we can import cell type labels so that we can make biologically relevant comparisons, and we can also do differential gene testing in treatment versus control or disease versus healthy samples of the same cell types. Now, all of these steps can be performed using a bunch of different available pipelines, but the most commonly used two are ScanPy, which is Python-based, and Seurat, which is R-based. Since Seurat is very user-friendly and there's a lot of visualization tools available in Seurat that need to be handwritten for some of ScanPy, this video today is going to focus mostly on Seurat. All right, so we're going to expand a little bit on each of those steps in the workflow by giving you some examples of what we view as standard single-cell analyses. More information on each of these analyses and figures can be found on the Seurat website. So to kick us off, before you combine many data samples together into one analyses, it's important to look at your raw counts table. So on the left here, we have an example of some nice raw data. We're plotting the number of counts per cell versus the number of features or genes that have expression in each cell. And we see that there is a very high correlation between those two factors. On the right, we see some quote-unquote poor raw counts data, where there's some dead cells at the bottom that have very low number of counts and even lower number of features, and at about 2,000 features, we see this forking effect. There's a split in all of our samples where the number of counts increases, but the number of features does not. This could be indicative of cells that were damaged or doublet cells where more than one cell was combined per well before library preparation went through. This doesn't mean that this data is useless. It only means that additional QC might need to be performed. So once we perform our QC and some of our normalization, we can look at what are the most variable genes across all of your cell types, regardless of what sample they came from or what potential tissue they belong to. And if we were to see that our top 10 genes labeled here are all mitochondrial genes or all ribosomal genes, then we would be a little concerned that those are the most variable features, and we might want to go back and relook at our QC and normalization. So it's a good check to make. So these figures probably look very familiar to anyone who's read any single-cell papers. Nearly all of them include some sort of two-dimensional single-cell cluster map. And so on the left is our default, where the clusters are numbered and colored by their cluster generated by Seurat. On the right, what we've done is recolor that exact same diagram based on some metadata features. And here we're looking at the tissue of origin, so subdural versus dural tissue. And we can see that the data isn't necessarily clustering by location, but probably more by cell function. So you could also color this by treatment versus control, or biological replicate number, or age of your patient, whichever factor is most relevant to you. And this is a very standard map that you'll see quite typically. Once we establish our 2D cluster map, we can use that and overlay expression of genes of interest. So in this case, we're looking at microglia-specific markers. And we can also view this data as violin plots of expression per cluster by cell. And you can see that when we look at the violin plot, we get different data than we do when we look at our feature map. So cluster 9 of AIF1, for instance, does have a small proportion of cells that express AIF1, but you cannot see that in the graph above. So once you've taken a target or cell population-based approach, it's also useful to take a discovery-driven approach to your single-cell analysis, which is where this heat map comes in. Here we're just looking unbiasedly at the top 5 most differentially expressed genes per cluster in the top 500 cells per cluster that have the most variable gene expression in them. And we can see that a lot of these markers are present in multiple clusters. So for instance, FEV in cluster 0 is also very highly expressed in cluster 6. So that marker isn't sufficient to identify just that cluster. And in addition to all these graphical figures that we've seen, there's also a lot of charts and tables that are standard output for single-cell analysis, such as charts of log-fold change expression across clusters, whether grouped by metadata features or by just their numerical cluster number, with various cutoffs, and also the cell counts per cluster. So like I said, if you want to get more information on any of these standard figures, there are a lot of tutorials and vignettes for Seurat available on the Seurat website and elsewhere. So we want to move on now from those typical figures to some of the expanded tools that you can use to improve your single-cell analysis. When doing single-cell analysis, the most important thing we can do is optimize our cell clusterings. There are multiple steps to this process, one of which is to do a principal component analysis. So after we choose our most variable feature genes, we're going to run a principal component analysis to reduce the dimensionality of our data set and feed a certain number of those principal components into our cluster generation algorithm. So how do we choose how many components to pick? Usually it's an educated guessing game where some bioinformaticians will start off with 10, some will start off with 25 principal components depending on the data set, and then by trial and error that number of components is adjusted. What we've done at Bisq is automate this process so that, building off of the jackstraw function, we choose only principal components that can account for a certain percentage of the variance seen in the data set and are significant, saving a lot of time and providing a statistical reasoning for why we've chosen certain components. So once we choose our principal components and we run our clustering algorithm, we next need to decide the setting for cluster resolution, or how strictly we want to subdivide populations of cells into clustering. And you can see with this cluster tree diagram that as we increase the cluster resolution, we get additional clusters being formed. And this lets you trace the lineage of those cluster splits. But why do we care? How do we make this biologically relevant so we can choose the appropriate cluster resolution? Well, one way is to take some of our marker genes of interest. In this case, we're looking for microglia, and we're using the microglia marker TREM2 and recolor our clusters based on average expression of TREM2. We can see that cluster 0 and cluster 3 at 0.4 resolution are our highest expressing TREM2 clusters, and that after resolution 0.8, both of those clusters are split into additional clusters. So we might choose our optimum cluster resolution as 0.6. We might also run this for multiple markers for our cell type of interest, or maybe just for our target gene of interest. The tricky part about these cell cluster diagrams is that we're trying to plot a three-dimensional map in 2D space. And so to improve upon this, we can also overlay cell density contour lines onto our two-dimensional plot to see which clusters are the most tightly packed and which might actually be more dispersed that our 2D graph doesn't necessarily show. In addition to combining multiple single cell data sets from different sequencing platforms, recently there have been tools developed to allow integration of multiple data modalities, such as incorporating epigenetic information from ATAC-seq. And this combination in the same space allows you to perform detailed co-expression analysis of different cell types. So let's say you have a data set that contains many cell populations, but you're not sure what the makeup of your single cell data set is. There are many different ways that you can actually import cell identities from reference single cell or other data sets. So on the left here, we've taken the Allen Brain Institute single cell reference data set and combined it with our microglia macrophage data set from previous slides. Unfortunately, while all the Allen Brain Institute data did already contain labels, only some of our cells, shown in gray, actually cluster with those identified clusterings. And what's easier to do instead is take our single cell cluster diagram of just our data set and actually transfer in labels from this data set based on similarly expressed gene patterns in these clusters. So we can see that the bulk of our cells have been labeled as microglia or non-microglial cells. Another popular use for single-cell RNA-seq is to perform pseudotime analysis of stem cell lines or other differentiating tissues. Pseudotime analysis enables us to rank cells by their progress along a process of continuous change, i.e. a differentiation process. Here we're looking at the differentiation of the olfactory bulb. Performing pseudotime analysis also allows us to produce pseudotime ranking and extract out stage-specific markers, which may have been covered up in bulk analyses. For example, a gene specific for a rare and unstable intermediate stage can now be validated in a differentiation assay. In addition to pseudotime analyses, gene regulatory networks can also be constructed from single-cell data. Now, typically you hear about GO and KEGG pathway networks, but today we're also going to talk to you about using the scenic functions. So these networks all group genes into co-regulated modules, which can show specific activity in a given condition, treatment versus control across differentiation stages, or in the case of this example, by cell type. So highly active modules for the scenic package are shown in black, and it is clear that this technique is a powerful hypothesis generating tool for driving networks of a given cell type or process. In addition to all of these visual outputs and their corresponding data tables, we can also generate additional useful data table outputs, such as looking at not only what genes are differentially expressed, but what percentage of the total cells per cluster express that particular marker. This is especially crucial for cell line data and brain data sets that contain differentiating or highly diverse cell populations respectively. Our goal at BISC is to allow you to be confident that you are fully utilizing your single cell data by optimizing not only the parameters that we use for generating your clusters, but also the visualizations that are used for your specific data sets and automating whatever steps we can to make this easily repeatable. We also have experience generating customized features such as graphic user interfaces like our Shinies, etc., for you to be able to interact with your data on your own without having to just stare at an Excel sheet. At BISC, we like to work as an extension of your team. And if you have any additional questions on any of the slides that you've seen here today, please feel free and reach out to us. Thank you so much.